Welcome to Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health, where we bring evidence, experience, and perspective to make sense of today's leading health challenges. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes.
Hey listeners, it's Lindsay Smith Rogers. Today, a new test for Lyme disease developed by high school students. I'm joined by Sankalp Yeleti, a high school senior at Lambert High School in Georgia, and Nicole Baumgarth, director of the Lyme and Tickborne Diseases Research and Education Institute at the Johns Hopkins Bloomberg School of Public Health. We talk about how Sankalp and his classmates, through Lambert High's world-renowned iGEM program, developed a rapid CRISPR-based diagnostic test for Lyme disease. We also talk about how Sankalp and his team connected with Professor Baumgarth and what this innovation can mean not just for Lyme, but other infections as well. Let's listen. Sankalp Yeleti and Nicole Baumgarth, thank you so much for joining us on Public Health on Call. To start us off, I'm going to have you each introduce yourselves. So, Nicole Baumgarth, let's start with you.
Yeah, my name is Nicole Baumgarth. I'm the director of the Lyme and Tickborne Diseases Research and Education Institute at the Bloomberg School of Public Health at Johns Hopkins University.
And Sankalp, let's go to you.
My name is Sankalp Yeleti. I'm a senior at Lambert High School, and throughout 2025, I was a member of the Lambert iGEM team, where we developed a diagnostic for Lyme disease.
And so we have you both on the podcast today because you worked together in some capacity on this project. And so, Sankalp, would you walk us through how you connected initially?
Yeah. So initially, what we wanted to do with our diagnostic was use PCR so that way we could directly detect Lyme disease and the DNA that it leaves behind in the bloodstream. And when we were looking into this, we wanted to see how feasible it would be. So we wanted to reach out to Dr. Baumgarth. And after being able to talk to her, she kind of helped rethink the way and the direction our project took. So while we wanted to work with PCR and directly with the DNA that this bacteria would leave behind, she kind of steered us more towards the direction of using outer surface proteins. So proteins that the Borrelia bacteria would shed into the bloodstream, and then testing for that and building our entire project on her advice.
And so we're going to get into a lot more about how your high school team developed a test for Lyme disease using CRISPR. But before we get into that, I think it would be helpful if, Nicole, you could talk about the general landscape of Lyme disease testing.
Yeah, Lyme disease testing, unfortunately, currently relies on a very old and very trying test. And in fact, it's two tests. The CDC requires what is called a two-tier test. That means two different methods of measuring antibodies, which are these proteins that the body develops specifically against the infection when we have been infected. So unfortunately, development of antibodies always takes time. And what we don't have when we get bitten by a tick that carries the Lyme disease bug called Borrelia burgdorferi is time. The biggest risk of developing long-term symptoms and ongoing symptoms after Lyme disease is actually having a delayed diagnosis and therefore delayed treatment. So measuring antigen is really a much faster way because we don't have to wait until the immune response kicks in.
Yeah, I remember when I was diagnosed with Lyme disease, it was because I still had the rash, which I think is a crucial time in the process, right? That's early on.
Correct. Yeah. That's actually a good thing. If you have the rash, you're very lucky because it's a telltale sign of having the disease.
But other than that, you've got to wait for these antibodies to develop. And again, that doesn't give enough time for the medication that you can take for Lyme disease to really do its job. So, Sankalp, your team was looking for a problem to solve, and this was a problem. So tell us about how you actually developed this test.
Yeah, so after we initially heard about these outer surface proteins, we decided to go ahead and we wanted to develop a full pipeline to be able to detect for these proteins. And this was a process of a couple months of trying to research and put together the entire thing. But essentially what we do is we can find and detect that protein as little as 2 days post-infection. We start with a process called PDL, which is proximity-dependent ligation, and researchers at the end of 2024 released aptamers that were specific to the CSPZ protein, which is what we wanted to detect for. So these aptamers bind to our CSPZ protein and they turn it into an actual DNA product that we can use. So then we use this DNA product and we use something else called RPA, which is recombinase polymerase amplification. So it's very similar to PCR, but we wanted to make our project point of care. So this allows us to do it isothermally. So that way people in rural areas, people to access without all of the equipment that they need, can still use it. And this amplifies our DNA. And then our last step uses CRISPR-Cas12 and a lateral flow assay. So a paper-based test strip, super similar to what you see with COVID tests, will then give you 2 lines if you're test positive for Lyme disease, or 1 line if you test negative for Lyme disease.
And what are you actually testing here? Is it blood? Is it spit? What are you looking for?
Yeah, so we would be testing blood, and then we wanted to validate this because testing in the lab isn't really something that gives us any information. So what we did is we got food grade bovine blood, so cow's blood, and we were able to spike it with the protein, and we could use our entire process on this, and we saw that it worked with actual blood.
That's fascinating. And so tell us, initially when your team was looking for this problem to solve, why did you choose Lyme disease as the target for this work?
Yeah, so every year we spend a couple of months, and all we do for those couple of months is just think of any idea that we could possibly think of. So as we were looking through diseases, the one that we came up with first was Rocky Mountain spotted fever, which is a tick-borne disease in the United States. But after a little bit more research, we found it affects 12 people a year. So it wasn't the scope of a project we were quite looking for. And then as we looked into other tick-borne diseases, we stumbled upon Lyme disease, not stumbled upon. It's very famous, but after we were able to look into this disease and talk to researchers, we saw this is a problem that we could actually have an impact on.
Yeah, and so you've essentially, as a high school team, developed a blood test to check for Lyme disease very early on in the infection process. What will happen now that you've developed this test? Could it be used for other diseases?
Yeah, so along with our actual pipelines like the PDL, RPA things, we developed a software to help streamline that process. And the idea is that if you have any blood-based biomarker, you could take this entire process and adapt it to that. So this would be work as a diagnostic for essentially any disease that leaves something behind in the blood.
And so going to you, Nicole, this is a pretty remarkable innovation. I imagine you don't get many high schoolers reaching out to you with an idea like this.
Yeah, I get quite a few emails from high schoolers wanting to work in my lab over the summer. And unfortunately, because we're working with infectious agents, the paperwork is just so large that that's very difficult. So I was very keen when I got an email from this group, and they seemed to really know what they were talking about in the email. They had very specific questions to exchange with them, so and Zoom with them and find out a little more. I'm happy to hear that I had some impact on what they were doing, but I was really impressed because I had maybe 3 Zoom calls with them, and it was really, they were informed and asked me very directed questions, and I did the best I could to give my answers, and then they went away. 2 Zoom calls later, they sent me a video with the entire process. The techniques that Sankalp was describing, mentioning, are really state-of-the-art techniques that are being used, and stringing them together, not just to detect something, but to detect something in a way that would eventually, if it worked out, be point of care is what is so important. The speed of diagnostic is really important, identifying it ideally right in the doctor's office. If you have the rush, that's great, that bullseye rush that is so specific for Lyme disease. But if you don't have the rush, or particular if you're an individual with darker skin, the rush is actually very hard to see, and it is actually different. Then having this point of care diagnostic is really what is so critical.
And do you think, is the industry ready for something like this?
Well, I think the industry would very much like a test for Lyme disease. It is not a rare disease, as Sankalp pointed out. It is the most common tick-borne disease in the country. Just by estimates, we have about 500,000 people or so every year that get infected, and way more people are actually being bitten by ticks and would like to know whether they're actually starting to get symptoms. So being able to detect the bug quickly after it is transmitted from the tick would be a change. And yes, the only thing we currently have is an antibody-based test, and that test is often negative, even on individuals that have, particularly early on when you have the bullseye rush, being seropositive is actually very unusual. and then you're usually getting treated with antibiotics. And what happens, because now you get rid of the bug, you actually often never seroconvert, you never make antibodies. So you'll never be totally sure that you actually had Lyme disease. So measuring the antigen, the way that this high school's team was going after it, is really the holy grail of Lyme disease diagnostic.
Well, and it's fantastic that, you working for the university have been so generous with your time and your work. But Sankalp, going to you, what's next for you?
Personally, I'm going to be going to NYU this fall to study biomolecular science. But for the project, our goal was to hopefully make this something other people could build upon. So we're trying to get all of our work published, and this will let other Lyme disease researchers hopefully use our work for something else.
Well, this is really remarkable. So Sankalp Yeleti and Nicole Baumgarth, thank you so much for joining us on Public Health on Call.
Thanks for having us.
Thank you so much.
Public Health on Call is a podcast from the Johns Hopkins Bloomberg School of Public Health, produced by Joshua Sharfstein, Lindsay Smith Rogers, Stephanie Desmond, and Rachel Walsh. Audio production by Michael Bonfils, Matthew Martin, and Philip Porter, with support from J.B. Arbogast, Spencer Greer, and Chip Hickey. Distribution and social media by Rachel Walsh, with support from Nick Moran and Brianna Iacovelli. Follow us on Instagram and Blue Sky at Public Health Pod for more public health takes and special content. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Thank you for listening.